So what I wanted to do was give a bit of an overview of some connecting themes that I've been working on, and maybe it's sort of like a transition type seminar where I'll sort of talk about work that sort of was finished up at Pioneer, and I think how that opens up a lot of opportunities for broadening the whole concepts of prediction for scaling or increasing the scale of a lot of activities in agriculture to meet some of the challenges if you think about some of the targets that we have for this century. And so what I'm going to talk a bit about as a foundational component is maybe the genomic prediction methodology that some people might be very familiar with, and how we might extend that by bringing in a lot of the information from different types of biological models with crop growth models, integrating information about how environments influence performance of traits and how traits might work together in different environments to influence outcomes such as yield and yield stability across environments. And so that's sort of this interface and extension of genomic prediction using crop growth models. And first of all, I'll start off by just acknowledging that this is sort of work that I'd been doing over a number of years with very large teams and a lot of other people. So I'm just recognizing some of my colleagues that I sort of had long-standing relationships with at Pioneer, and you'll hear about some of their work here. And also the long-term connection with Graham Hammer and Scott Chapman here, based in Australia. So it's sort of, I guess, just recognizing those long-term relationships. And so while I might spend a lot of time talking about some of the extensions of genomic prediction methods using crop growth models, what I want to do is sort of position that into some broader prediction questions and hopefully start to highlight some of the opportunities beyond just sort of working at prediction inside breeding programs. And I think that this sort of talks to the whole question of how do you align your capabilities, your prediction capabilities, with what are the important or the right questions that you actually want to ask. And so you don't end up with answers such as 42 for anybody that's familiar with the Hitchhiker's Guide to the Galaxy. And so, you know, what I've been trying to do is align some of the prediction capabilities with different types of questions that different disciplines ask. So breeders' questions, agronomist questions, and growers' questions. And this sort of starts to help you to think about what are the prediction needs from end to end or start to finish, from product creation using all of our genetics and physiological knowledge, right through to helping growers to optimize new genetics out of breeding programs with management practices across a diverse set of environments, so enabling end-to-end prediction. And I'm going to talk about this in context, given that I've been a maize breeder for about the last 15 years at Pioneer, and was involved in developing commercial products that were being grown by farmers in the US, but also some of the hybrids that I was responsible for are now being grown in Europe, and the germplasms being used in a lot of other countries around the world. So you're not talking about something that hasn't made progress. You're actually talking about building on top of something that's been undergoing this really long-term genetic improvement that's very successful, as Graham mentioned, and it's a highly competitive type system. So, you know, what we're trying to do is not just predict something where there's nothing. You're actually trying to predict the extensions beyond what's already very successful. So that makes it a pretty challenging type of exercise. So this is just a graph just charting out a series of very successful hybrids that Pioneer commercialized in the Central Corn Belt in the US, and looking at them plotted by their year of release in a set of different types of environments, from very favorable environments up at the top here, right through to severely stressed environments where you've actually removed a lot of access to water, nitrogen, important resources obviously for improved performance. So if we think about genetic gain and where genomic prediction fits in, just to sort of orient everybody, If you're not familiar with that, if you think about sort of a very classical breeding program where we were back in the 1990s, I mean, a lot of what we were doing was really driven around what you could empirically grow, evaluate, and phenotype in the field. But a lot of the ideas for using fingerprinting or molecular technologies to fingerprint genotypes in breeding programs started to develop back then. But you were still really dependent on improving your capabilities in the field, your plotsmanship, so you could actually phenotype really well, improve the heritability, and use that to whatever scale you could actually get to, and how quickly you could turn around the cycles of your breeding program in time to drive genetic gain. And so this is just an aerial picture of sort of a lot of the stages from one of the locations that my maize breeding program actually operated at. So there's a cartoon sort of on the side here that just sort of highlights how a typical sort of competitive mid-maize commercial breeding program operates. And if you're familiar with different breeding methodologies, it's basically what we would call an open reciprocal recurrent selection program, where we're actually driving pedigree breeding on both sides, two heterotic groups. We're trying to drive pedigree breeding to improve the females and the males. They have to complement each other to ultimately produce improved inbreds that work together to produce improved hybrids. So what you're looking at here is just a whole series of experiments that representing these different stages of testing actually being conducted at one location, where we could manipulate water or nitrogen availability to go from severely stressed right through to very high yielding environments, all at one location. So what we are able to do and what we did become able to do was, as well as measuring all these traits, we could actually fingerprint everything that was actually inside this program with a sequence of molecular markers. And you could do that at different levels of density across the genome. And typically in a breeding program, we would align that with the levels of linkage disequilibrium that we were actually dealing with at the different stages of the breeding program, depending on how much recombination we were actually driving at the different stages, or how we were actually making those crosses. This enabled us to take any one of those individuals in here and for a series of positions across the genome. So if you've got a whole lot of the inbreds that are being tested in hybrid combination in here, you're looking at a position on the genome, and you're just looking at a fingerprint that's converted into a color graphic that just shows different segments that have come from key founders earlier in the history of the pedigree breeding program, and how they've been passed down and recombined to create the different inbreds that are actually under test in here. Just so you're basically turning the sequence information from identity and state in terms of the SNP information into identity by descent, tracing back to the key founders. So you can start to talk about these regions in terms of their origins throughout the breeding history. So what we then wanted to be able to do is obviously take this information, this fingerprint information, combine it with this phenotypic information here, and build a genetic correlation basically between the sequence polymorphisms and the phenotypic variation for these traits. So basically the conventional historical breeder's equation becomes a form something like this, where you're actually looking at the genetic correlation between the fingerprints and the additive effects or the breeding values of the alleles or the regions in the genome that you're actually fingerprinting, and thinking about the additive genetic diversity that you're working. And in this case, obviously, the additive genetic diversity on both sides of the heterotic pools and how they come together to make the genotype. So that's the basic engine. So you've got all this phenotyping, you can genotype. You've sort of got this basic framework, and we've got association methods for associating these polymorphisms with these this variation actually up here. And so this enables you, because we could genotype things much more cheaply than we could phenotype, we could actually take our conventional program and actually increase it by orders of magnitude. So basically you have this huge scaled up breeding program where you've got an engine that's run around the phenotyping that you're actually doing and the genotyping. And then any genotype any individual that's genotyped anywhere in the world where you've got a fingerprint of that, where you've got that allelic polymorphism, you can actually make a prediction from the effects that are associated with these polymorphisms from your training data set for any individual and decide whether or not you actually want to bring that into your breeding program. So the whole thing becomes very high scale. And so we talked a lot about the history of the development of this methodology as it came into being in a commercial breeding program in a review article, and it sort of just charted out some of the key components that came together. Talked a little bit about the history of the germplasm. This is just the germplasm galaxy where we're looking at pedigree diagrams where you've got the founders in the middle, and then you can follow these pedigree diagrams out to the fringe where these are the elite individuals out here. This would be for the female side. Then you can follow the same diagram out here to the elite individuals that would be out on the male side. And then these are the elements that I talked about before. And then this basic machinery here where we're basically trying to use this information from the phenotypes and the genotypes using some algorithmic approach to estimate genetic parameters that we can then use to actually make the predictions on things that we've fingerprinted. So that's basically a summary. And just to give you an example of the types of genetic correlations we would be able to get between those polymorphisms and the total genotypic value of individuals. This is an example where there was an experiment that was run over 2 different years, and we used 1 year as a training data set for the next year for a range of different traits that just gave you this range and heritability level down here. So things that were sort of, if you like, harder to measure had lower heritability. Things that were easier to measure would have this higher heritability here. And you'll see that there are 4 bars, and these 4 bars are color-coded here. The number on the outside here just indicates the number of hybrids that we're actually making predictions for. And just to quickly orientate this, the real just means that 97 of those hybrids were grown in both years. So you could actually make a prediction based on a training data set in 1 year to the same hybrid the next year and predict how would that hybrid actually perform next year. The T by T just means that the parents of that hybrid as inbreds were actually included and they were in some hybrid combinations in the first year, but that hybrid combination wasn't actually made, so they're tested by tested inbreds, but a new hybrid combination was actually made in the following year. So it's a new hybrid, but the inbreds, the parents of that hybrid, were part of the set of hybrids that were evaluated in the previous year. Then tested by untested, it sort of gets a little bit more interesting, where we've actually brought in completely untested inbreds that have only been fingerprinted somewhere. They've been brought into the data set and they've been combined with one of the tested inbreds. And then the U by U in this case is 2 completely new inbreds that were fingerprinted somewhere, and they were brought into this experiment, created as a new hybrid, and then grown next year. And the interesting thing you can actually see is that as you look down this set of traits, the different heritability levels, you can see for all of these hybrid types, prediction actually works, where you've got the genotyping done at the right level for the linkage disequilibrium you're dealing with, and you've got adequate training data sets that are suitable for getting estimates of these effects that are representative of the environments in the future year. So the bottom line is it works. Okay, so we wanted to build on this, and that's what I'm interested in. So that's the basic framework. It works, and I would say that most commercial breeding programs today, certainly the large major crop commercial breeding programs, are operating with this as a core part of how they actually do business today. So if anyone says, and a lot of people ask this question, does this work? Will we ever have products out there? I'd say from about 2010, a lot of the commercial products, certainly in maize, that are used in agriculture throughout the US and sort of more so in the other countries, have been developed through this sort of algorithmic approach. And so what I'm interested in extending this prediction methodology is how can we actually go beyond just sort of predicting to these traits and building training data sets and actually better use the biological knowledge to actually go further than what we can at the moment. And so just building on this sort of long-term picture, what we started to try and do, and I was really focused on developing products with stability of performance that could handle stress environments, water-limited environments, so drought was a major target of my breeding program. So we were building a lot of biological knowledge of what it takes for a maize hybrid to actually perform in water-limited environments. And so part of that research was just looking at this long-term historical picture and asking questions like, well, what's actually changed over time? So if these things are better performing under drought, water-limited conditions, is it because they're getting more water? Or are they using it more efficiently? So we started asking questions like that. And the obvious one and the prevailing hypothesis was that, well, they're more drought tolerant because they're getting more water. And certainly there is an aspect of that. If you go and look at the really early hybrids from the sort of 1930s, the double cross hybrids, and compare them to the single cross hybrids, if you actually look in a soil profile and compare the newer hybrids, the single cross, to the older double cross hybrids, and look at the rate of water use per sort of volume of soil, you can actually see that the older hybrids, the double cross hybrids, were certainly drawing out water more rapidly from the upper profiles and becoming less effective at getting sort of the water out of the volumes of soil at the lower profile, whereas the single cross hybrids were actually relying less on this upper water and sort of actually starting to use water more effectively over the whole soil profile. And so there was an element of that, so we could actually get at some of the different patterns of water use. But then when we actually went into the single cross hybrids, the more modern hybrids, and this was part of the PhD work at University of Queensland by Andreas Reyes, and we looked at the total water capture for hybrids released from the 1960s to these modern hybrids. We didn't see that there were differences in total amount of water capture. But if you actually went into some of these very sort of modern elite hybrids here, and those that we knew differed in performance under water-limited conditions, we actually started to see that there were really differences in, even though there weren't necessarily differences in total water use, there were changes in the timing and pattern of water use over time. And so this was work that was then done by Charlie Messina and his team, where we actually compared what became a commercial Aquamax hybrid, drought-tolerant hybrid, versus something that was quite sensitive to drought. We could actually see, if you looked at the pattern of water use out of the soil profile, that the more drought-tolerant hybrid in this case was actually using water at a slightly lower rate than the thing that was a little bit more sensitive. And in this water-limited environment experiment here, we saw a difference in water availability at flowering time that amounted to about an inch of water, 25 millimeters of water availability at flowering time. So hold on to that number and I'll show you how significant that might actually be. So this was an experiment where we then went to test some of these ideas. This is actually a large plot demonstration experiment that's been cut out so that we could actually look at the plots. And we did this at two of our managed drought and managed environment stations where we could manage drought. And I'm going to look at this experiment that was done in Chile at the Veluco station south of Santiago in a bit more detail, where we've used sort of managed irrigation to impose a sort of a favorable environment right through to our flowering stress environment here side by side. And we then have different sets of hybrids in this comparison experiment here. And so we take this experiment and sort of will actually husk back some of the ears in here and actually look at what happened. And so now we've just got a direct comparison between one of these drought-tolerant hybrids versus one of the more sort of sensitive type hybrids. In the well-watered, fully irrigated treatment, the grain filling stress and the flowering stress treatment that you saw side by side. And it doesn't you don't need a combine or a measurement approach just to see the visual effect here. In the favorable environment, there was sort of a slight difference. The yield potential of the hybrid that had been selected for better stability actually was a bit better than the other more sensitive hybrid. Under grain filling stress, you start to see a bit of a difference between the drought tolerant hybrid and the sensitive. You look around in here and you'll see a few ears without many kernels on them. And then when you go into the severe flowering stress, you sort of see a difference that's really quite dramatic, where the sensitive hybrid has gone completely barren, basically. And so this is sort of what a difference in 25 millimeters or less of water can do when you're actually caught at flowering time. It can scale to basically complete crop failure to something that's like 10 or more tons per hectare if you've actually selected for the right combination of traits. So that's sort of the scale that we needed to get to. And basically that's what's happening throughout all of these stages of my breeding program. We're actually managing the inputs into all of these experiments so that we can actually ensure that we're seeing differences in patterns of water use that are associated with these differences in rate of transpiration. We were convincing ourselves by including a whole lot of sensitive and tolerant checks in all these different stages that we could consistently separate the tolerant from the sensitive checks inside these experiments. And then this would determine, out of all the experiments that we were running, what were the quality data sets that would actually go into the training data set so that we could actually drive the genomic prediction as different breakouts for these different types of environments using these managed environment capabilities. And then you could do a whole lot of different analyses looking at sort of how big a training data set had to be, the composition in terms of populations, germplasm diversity that actually needed to be in there. And if we were only looking at yield as an endpoint, this gives you an idea of how we could manipulate the size of our training data set, for example, to manipulate the prediction accuracy that we were getting that was part of that genomic prediction equation that I showed you earlier. And so the two different colors are just to show you here. These are data sets where we're actually really focusing on how the checks were separated in these different experiments. And these are the new entries that haven't been tested where we're actually trying to predict their performance. Okay? So again, with this level of predictability as a breeder, I know that I could make this work in a breeding program. And this level of predictive accuracy, everybody says, well, it'd be great if it was better. I'd say, yeah, but it's better than zero, so I know I can do something. And if anybody has tried to do drought in sort of your target environments where storms float around and all sorts of other things happen, getting to this level of predictability is just about impossible. The other thing that's really important, which goes down to the time part of how quickly you can turn this around, because we were using northern and southern hemisphere, I could actually turn around 2 cycles of testing each year, working between the northern and the southern hemisphere to speed the whole thing up. So there's all of those elements that are actually going on that you're integrating to make this an effective breeding program. And so that sort of is all the background, and this sort of gets to this whole question of how can we start to leverage all of this extra biological information and understanding and integrate that into the genomic prediction methods. And so even if I don't get through this or I don't explain it very well, you can sort of look up this sort of recent paper that I think is still in press, but you can look at it online, where we sort of talked about some of the integration of a lot of this knowledge using the crop growth model into the genomic prediction. So I'm just going to quickly go through some of the ideas and highlights that are sort of embedded in this paper. So I talked a bit about what genomic prediction is. If I didn't do a good job on that, I apologize, but please ask questions. So I guess I keep asking myself, or I keep asking Graham Hammer anyway, what's a crop growth model? And he says, well, it's a whole bunch of equations that capture all of your knowledge and we can make predictions. And he says it in such a convincing way. I usually say, oh, that's really great. And then I walk away and I think, yeah, I'm not sure that I got all that, but anyway. Then I went back to him and I said, Graham, you know, I've been working on these network ideas for about 20 years or so, and I said, I think your crop growth model is just a network. And he says, what do you mean? So this was my first attempt to try and say to Graham, well, you sort of give me this developmental growth and development sort of process, and you tell me there are all these equations, but if I draw arrows on this thing, to me this just looks like a network diagram. So it's another network diagram. And he says, yeah, I'm not sure if you're right, but, and so you know what it means when someone says but, what they don't think much about what was said before, and then we kept talking anyway. And so then the next question was, I said, well, and I talked to Charlie about this, and he says, yeah, well, we can reorder this diagram, and it looks a little bit more like a network diagram. And so I said, yeah, it does actually, and then you can start to think about each of these arrows has its own equation, and they all have coefficients in them, and they relate to growth and development processes and traits and environmental inputs. So basically, it is a network diagram. So can I connect this to quantitative genetics and all the work that I'd been doing on trying to recast quantitative genetics from the infinitesimal model into network theory? And so I said, okay, I'm sort of going to think about this a bit harder, and how could we actually do this? And sort of just to orient you, if you do a sort of a quick tour of 100 years of quantitative genetics, if you start off with what is a finite locus model, well, most people would think about that as a whole series of independent genes that know nothing about each other, and then they don't speak to each other, but they've got their own effects, and you can add them up to predict the value of a genotype or an individual. And so in terms of a network diagram, sort of taking this idea here, I've just drawn what that network looks like. So it's a set of genes that just sit there. They're nodes that don't speak to each other. And if you were to think about that in terms of network theory and topology, it's basically an adjacency matrix where all you're doing is putting effects on these diagonals, and there are no off-diagonal terms. And so then I said, well, what if these things actually do talk to each other? And so these are the types of networks that we can actually discover when we look at how genes are actually operating in networks, or as we map traits and look at how QTL and genes actually interact with each other. And basically these sort of arrows or connection, these edges, actually start to connect some of these nodes. So basically what you're doing is you're filling in some of these off-diagonal terms in the adjacency matrix. And anybody that's familiar with sort of statistical theory and sort of topology starts to realize that all of the machinery of variance covariance matrices and sort of adjacency matrices and so forth sort of is not that far away from thinking about a crop growth model and quantitative genetics. And then you realize when you map a trait, well, you don't really find all of the genes of the QTL. You only find some of them, and there's this dark area here, so you don't really know all of the things that are going on. You may have this part of the picture, but you've sort of missed out on this part of the picture here. So there's interactions with backgrounds, because some of these things you found are still interacting with things that you don't find, so you always get these sort of QTL by background effects. And so those were some of the models that we actually had, and we sort of introduced this theory and talked about it sort of in this paper back in 2005, and just showed how the classical quantitative genetics models could be recast in terms of this sort of network theory, which was sort of developing independently of quantitative genetics largely. And also you can have genotype by environment interaction, where some of these genes are switched on and off depending on their environmental inputs, which is sort of very similar to sort of those environmental inputs that are affecting these coefficients inside the crop growth model. So, yep, said that. And then with Charlie, well, I pressed the wrong button there. With Charlie, we just said, well, we can take those equations on those edges that are connecting the nodes, and actually start to think about the coefficients and how they could connect to these underlying gene network models, which is what we did. So we took the publication by Francois Tardieu and his team, where he was looking at the output trait leaf elongation rate. He had a biophysical model of this in relation to the meristem temperature, vapor pressure deficit, and soil water potential. Their coefficients, this was this basic equation. You could map these coefficients and terms in this equation, find QTL. Those QTL could work independently, or they could easily just be reinterpreted in terms of these nodes and edges on network. So what we could do is we could say, well, each one of these arrows has a whole series of equations, and they've all got coefficients. And so if we could phenotype this and we had a reasonable model and we knew which traits were segregating in my breeding populations, we could actually start to use this machinery. And so what we're basically now doing is thinking about the underlying quantitative genetics of the traits that ultimately affect yield, but also how they're doing that through some of these intermediate traits. So could we actually make this all actually work? It would work if we had a model, we could find the QTL, and we could quantify their effects, we could phenotype, and we could do all of that. So that's what we set out to do. And this is where Frank Technow joined us, and so he had a lot of really interesting and really good ideas, and it ended up being, it's a very simple implementation that we've actually talked about in these papers. And so basically what we're doing is, instead of trying to do the associations between the polymorphisms and yield, we're actually going to use the associations between the sequence polymorphisms and things we can measure about these traits, and then use the crop growth model to go from these traits up to yield. And the principle is that there's a lot more interactions than we can sort of sort out by going directly from sequence to the yield and sort of how that varies across environments, versus some of these developmental traits which have more predictable responses across some of these environmental variables. So here's an example where we now have our training data sets. We want to know how we can use these training data sets to get effects for these polymorphisms, these genetic effects. Given all the data that we can actually generate, we've got a crop growth model which we can integrate into the likelihood function, so it becomes part of the link function, linking the sort of the underlying genetics to the traits, and then through the crop growth model ultimately to yield. And we can measure as sort of prior information also the traits that are varying in my particular populations, and use that to set up some starting values that we're going to sample from, sort of, and then use the prior in combination with this to say, well, does the sample actually match the data that you've actually got, and then use that through appropriate algorithms to actually build up the posterior distribution that we're going to sample for the effects that we're going to use for prediction. And so the underlying model is relatively straightforward for some of these underlying traits. You've basically got a predictor for the traits in the crop growth model. So this is limited transpiration. This is a measure of reproductive resiliency in maize that's related to that catastrophic difference that I showed you. We talk about it as minimum ear biomass. Canopy architecture, how it influences radiation efficiency, and canopy size, measured by the size of the leaves in those canopies. You've got an intercept, you've got a set of genotypic coefficients and effects that are related to these traits. And then the crop growth model knows how these traits vary across environments in relation to temperature and other variables. So from there, we sort of went through a series of proof of concept studies, and we wrote one of those up in a paper that was published in PLOS, one where we actually took an artificial simulated scenario with a nonlinear relationship between canopy size and yield for two environments, and actually showed that if we tried to do a direct prediction to yield that didn't have any mechanism for adjusting the effects of these QTL as you go across different environments, whereas we use the crop growth model as an intermediate in here with the whole genome prediction, we could actually do a better job of predicting yield. So we're sort of getting values in many cases that are above this one-to-one relationship. So we were actually doing better than just directly trying to predict yield using the data set, the training data set. Same training data set, but now we're using these intermediate variables. So that was all done in silico. Then became the Herculean task of, well, could I actually do this in one of my breeding programs in one of these drought environments in this managed environment station at Veluco? And so this is the result that we actually got for the first population we tried this on. where we had to get all of the inputs, we had to get the model right, we had to get the right traits, and basically, and into the same environment. So it was sort of like the simplest scenario that I could think of, but it was still in the real world. And what we could actually show was that, yes, indeed, if we had picked the right traits, we had a decent crop growth model, we got the right inputs, we could actually predict yield using the crop growth model. So in this case, here is a crop growth model actually doing your quantitative genetics and predicting the yield outcome. There's no actual mapping to yield. All we're doing is mapping to the intermediate traits. And the crop growth model is doing all the prediction of yield. So there's no mapping of yield in here. So this is sort of like the first time ever that a crop growth model had actually done the mapping of yield variation in a real breeding population. And it worked. So again, it comes down to the same basic rules. If you've got the right training data set, you know the traits, and you've got the integrating tool, the crop growth model, you can actually predict yield. So then the next bit was going back to this paper where we then started to work between sort of in silico work and then extending this from one population to multiple populations. And so this is an example of results where we were actually predicting to different environments for different populations. And you can see that in a number of these cases, we actually did better. In some cases, we didn't do as well. So there's no free lunch. It doesn't always work. But it does, on average, do a lot better than if you're just trying to predict the endpoint yield. So how did it actually operate? So the mechanical machinery is sort of explained inside that paper. It's really quite a simple framework. I hope I've convinced you of that. We're actually trying to predict yield, where the crop growth model is the link function. This is just Bayes A. This is just standard Bayes A for anybody that's familiar with genomic prediction, pointing to the traits that are the coefficients of some of these traits, so they're the edges inside the crop growth model. So we're mapping to those traits. We have environmental responsiveness that allows us to modify the allelic effects as you shift your environment, that then allows you to make modified predictions for yield with this framework. And this is just a residual variation out here because you've got to deal with error. So it's pretty straightforward. So this is sort of moving between the in silico into the real data set. This is sort of inspired by sort of 3 different years at one of our station locations in the Midwest. And we picked 2 challenging drought years, 2012 and 1988. And the reason we picked those, we also did this with 2010, which was there was no drought stress at this location. The reason we picked these 2 years was because everybody said, well, you can never predict what's going to happen in 2012 or 1988. No one could predict that. So we thought, well, it'd be interesting to see if we could actually come up with scenarios where that would be possible. And so this is just using an index of water availability. 1 means there's no water stress. 0 means basically there's no water available left to meet the demands of the maize canopy. And so this is just showing you sort of the patterns of development water deficit, in relation to development. This is the flowering time. So 2012 is the solid line, the dashed line in this graphic is 1988, just showing you that in 1988 we got a touch of flowering stress as well as grain filling stress, whereas 2012 was another drought year, but it was mostly grain filling stress that we actually got. And this is sort of simulating using the crop growth model, what we would expect for 4 traits we were interested in: the canopy size, this reproductive resiliency, radiation use efficiency, and then sort of breakpoint part of this limited transpiration again. As we actually increase the value of this trait, the canopy size, and this is sort of its relative effect on yield in the 3 different environments. And I'll start off, first of all, just orientating you. So this is the favorable environment, non-water limited, this dashed up here. So basically, as you're increasing canopy size, you're driving yield up. So bigger canopy, favorable environments, intercepting radiation, you can transpire, and sort of bigger canopies gave you sort of higher yield. Whereas in the water-limited environments, you can see in both of them, the tendency was to go down. So basically, the bigger canopies were using up water, and you tended to get into more trouble if you were too thirsty and you used up water in these water-limited environments. And then you can sort of look at the traces for minimum ear biomass, reproductive resiliency. If we look at the non-water limited environment, you can see that shifting that from a low value to a high value didn't have much effect. High value would be sort of less reproductive resiliency. So you've got to think about the scale in that sense. And I can go into those details. Whereas in the drought environments, this larger minimum ear biomass, you can see started to cost you something in the drought environments. And you can see that that was a more painful effect in the environment where we had the flowering stress. So that reproductive resiliency, going back to that graphic I showed you before. And then you can look at the same thing for radiation use efficiency. It's got a similar pattern, but more separation than as for the canopy size, so more efficient canopies. Basically, if you were higher radiation use efficiency, you were growing more and you were costing more water and getting into trouble in the water-limited environments, whereas it was good in the favorable environment. This limited transpiration, which regulated the timing of water during growth and development, a higher break point meant that you were less conservative, so you were burning up more water. In a favorable environment, burning water was good, but in a water-limited environment, burning up water was less desirable. So basically, that's the machinery we were capturing with the crop growth model. And then just to show you the differences in the predictions between trying to predict yield just using Bayes versus using the crop growth model as this intermediate link. And so here we're going to do a comparison between, in the first case, the water-limited environment in 1988, predicting to other genotypes but staying in the same environment. And if I highlight this, you can, oops, that wasn't what I meant to do. If we highlight that, you can see that both, in this case, the crop growth model and Bayes in the simulated example did equally well in the same environment, which was similar to what I showed you in that empirical example. The predictions were lower in the real world case than in this simulated example, but it's what you actually get. So you've managed to capture the machinery, the right traits, so you can actually make the prediction. Then if we go to the situation where we're predicting from one drought environment to the next, you can see that the crop growth model is a different type of drought, remember, actually did a little bit better than sort of just trying to predict to yield. And then it becomes much more dramatic when you actually try to go from the drought environment into the sort of favorable environment. Whereas in the drought environment, you get all of these effects associated with yield that aren't shifted as you go to the next environment. And the environment sort of says these traits really need to be in the other direction. And so you get this negative relationship between what you predict and what happens. But the crop growth model then modifies the value of the effects of these alleles to say, yeah, bigger canopy is going to be a disadvantage in this sort of water-limited environment or an advantage in this favorable environment, and it sort of makes that adjustment for the prediction on yield. And so with the crop growth model, you actually do a good job of predicting, whereas if you're just going to the endpoint trait, if you can't adjust for the shift environment, you actually don't do such a good job. And then sort of what you're really taking advantage of is sort of the ability in these different environments to get information about these different traits. So this is actually looking at the predictions to separate sets of genotypes, but for the intermediate traits. And what it shows you in the water-limited environments, you're getting some information about all of these different traits, whereas in the non-water limited environment, you can see you get a lot of information about canopy size, radiation use efficiency. You don't get much information about reproductive resiliency. You get different type of information about the limited transpiration. And then if you design a multi-environment trial testing system, you can actually get different information from the different environments, so that on average, you've got collectively more information about these different traits that you're actually using to parameterize the coefficients inside the crop growth model. And so you're now sort of using the mixture of your multi-environment testing system in the training data set with the crop growth model, the right traits, to parameterize the crop growth model with the QTL connected to the coefficients in the crop growth model to drive a lot of these predictions. And then this allowed me to actually go to a situation where I could use a set of populations to predict new populations in different environments, and in many cases do better than if I was just trying to go to drought environments and only trying to predict on the endpoint trait yield. So it's the sort of basic machinery. I'll just finish off with sort of one other point. So that gets you to the breeding point where we sort of have integrated the crop growth model. We can use that sort of in a lot of the early stages of the breeding program to sort of test and sort out all these potentially the new recombinants that you can create. And sort of what you're trying to do is build these products. But by the end of this testing system, you've sort of selected a few products that you're going to commercialize. The thing that you sort of realize as you're doing this is that you now parameterized a lot of the coefficients for these new genotypes around the crop growth model. So at the point of commercialization, you've already got the ability to predict how you expect these new genotypes to perform in many other environments. So you've actually parameterized your model by the point of commercialization. So down at this point at the end here, what you're now interested in doing is a fixed set of products and saying how will they perform in many other environments. And so this is sort of what Charlie Messina had been working on a lot with the crop growth model, was saying if you can get the coefficients for the crop growth model for these genotypes, you can actually predict the commercial product and all the new pre-commercial products that you're comparing against prior to commercialization. So you could do that by directly measuring all of the traits on these products in the right environments. But alternatively, if you've built those products through the history of a breeding program, at that point of commercialization, you can actually use the genomic prediction connected to the crop growth model to do all of that prediction as well. So it gets you to the point where at the commercialization stage, instead of sort of having to reparameterize all of the model, you've sort of seen these two genotypes. So this is just an example where we've actually done this. And you know that you've got this sensitivity in the stress environments, and you've got a difference in performance where there might be superior yield potential with this product. Could you actually go from this empirical situation that we can study, sort of do this type of prediction, but actually use the coefficients you've generated from the crop growth model to predict to all potential environments? So what we did for these two genotypes was take the parameterization, take all the historical weather data, the soils maps for the US, and actually went and predicted how we would expect those genotypes to compare to each other in every possible scenario on a 30 kilometer by 30 kilometer grid for 50 years in the North American Corn Belt. And what you can start to see is that you can see the overall target population of environment separation of these two hybrids. The interesting thing is you can see what often confuses us when we study pairs of hybrids, that there are environmental situations where the thing that can completely collapse can actually do a little bit better than your drought-tolerant hybrid, even when water limitations are actually reducing yield. And this can happen when you get a rainfall event just at the right time. So you're running into water deficit at flowering time, but then along comes a rainfall event. So your drought-tolerant hybrids being sort of holding back, being very conservative. Your sort of more high yield potential hybrids done, sort of been really aggressive, used up all the water, and then you get a big rainfall event that refills the profile, and it just goes on and actually yields better. So there's no mystery behind these things. They're not unrepeatable or undefinable genotype-by-environment interactions. You've just now pushed down to a biophysical interpretation of these genotype-by-environment interactions. So what I've tried to do is just use one paper out of many that I could have picked on, just sort of talked about trying to align our prediction capabilities around the types of questions that we're interested in, how in the maize world sort of we were able to connect this up and sort of just given you some examples of things that we've actually published on and talked about publicly. There's a lot more than this, obviously, in the background in the commercial world. But it sort of hopefully is an example that shows you that this can work if you sort of apply the same sort of principles. So what I'm sort of really interested in, sort of in this new role at UQ, is just thinking a bit more about the properties of the right types of models that we could use to build these biophysical link functions, and what types of information can we actually expose and use to understand genotype by management, by environment interactions, and what traits might be important, so build on the legacy of the work that's actually being done here. And think about ways in which we can extend our sort of growing capability to understand the genetic architecture of traits and start to fuse some of these models together to improve our levels of predictability. And it sort of then starts to help us to think about what are the true limits of predictability in these systems if we're trying to use prediction methods to scale our systems. So previously, I think most of us would have thought heritability was sort of like our upper limit, and it is in many respects an important consideration, but it may not be the only thing that we can think about, and we may be able to actually get beyond the heritability we observe in our target system if we can actually use some of this other information. And then I think that sort of can nest inside our broader context that sort of has been quite an inspirational discussion that I've managed to get involved in already with Christine Beveridge and a team that she's pulling together, where you can actually think about this application in agriculture as sort of what I've been talking about here as relatively short-range prediction type questions. But if you think about the overall landscape that we're trying to navigate in breeding programs, there's sort of this bigger landscape that we're actually trying to navigate across. And so how we move traits around from different sources of germplasm and so forth, I think this sort of gets into a bigger question that I'm sort of ultimately interested in, and that is learning how to navigate across performance landscapes. And so what velocity and directionality can you actually take? And I thought I was the only one that even asked this question, and most people laughed at me or thought I was crazy for actually posing this, but Christine actually sent me a review paper that I read this weekend, and in evolutionary genetics, they're actually talking about building flight simulators to actually navigate fitness landscapes. So they're already actually putting this into practice for thinking about evolutionary systems biology. So why wouldn't we be doing it for agriculture? And I think that sort of is a really exciting possibility for the future. So I'll leave it at that. Thank you very much for your attention, and I'm glad to be back.